Hi, Michelle Glass here, and welcome to episode 4 of our Chapter 3, Lecture 3 series. And our topic for this video are the proteins. So we are still working through our organic macromolecules. We've already discussed carbohydrates and lipids, and now our job is to look at these proteins. Now, proteins are super cool because these are the molecules that are responsible for so many jobs in our body. So we are going to start actually by talking about the different jobs of these proteins, and then from there we'll talk about protein structure. So when we talk about proteins being significant, because I'll stick my camera there, the proteins are determined by your DNA molecules. So when we talk about hereditary information and genes that are passed from, parent to offspring, what we're really talking about is the direction for what proteins to make and how to make them. So all of the things that we think about as hereditary traits are really just proteins that we're making. So it's like passing on the recipe book from parent to offspring to make these proteins. So super cool molecules. One of their important jobs is to catalyze our chemical reactions. And the type of proteins that are responsible for catalyzing chemical reactions are called enzymes. Now, we've talked a little bit about enzymes already, and we will have more discussion on enzymes as we go through the course. Remember, when we talk about a catalyst, we're talking about something that is involved in a chemical reaction, but it's not actually changed or used up by being part of that reaction. So we typically will see enzymes written above the arrow of our molecular equations. Enzymes are biological catalysts, and the significance of these is that they are making sure that our chemical reactions can take place in our cells quick, but also safely. So we don't want big combustions of energy given off in our cells, and we don't want them to take, years for our chemical reactions to occur. And it's the job then of the enzyme to allow that to happen rapidly but safely inside of our cells. And what we'll see is that essentially, in order for a chemical reaction to occur in the cell, you have to have the appropriate enzyme. So cells can control what chemical reactions can happen by controlling which enzymes are being produced and which enzymes are turned on or active. So enzymes are responsible for controlling cells' metabolism. Remember, metabolism is the term that means all chemical reactions that are occurring in the cell. So any anabolic or catabolic reactions are being coordinated by enzymes. So again, in order for reaction A to happen, you have to have enzyme A available. And so it's up to the cell to produce and turn on that enzyme. Again, I hope you're kind of getting this idea of specificity. Enzymes are highly specific. They are going to fit together with their substrate. The substrate is the molecule that's going to fit in with the enzyme. And it's like, match made in heaven. Only this particular substrate should fit with this particular enzyme. It's often described as a lock and key model. So just like your house key only opens up your door, then only this enzyme will be able to fit with this particular substrate. When they fit together, they're lowering what we call the energy of activation. So we've mentioned that in an earlier video. We'll talk about it again in the future. That's really talking about the energy that's required to get the reaction to happen. So again, this is speeding up the chemical reaction. So it's not taking years to happen. It can happen, in a timely fashion, but still happen safely inside of our cell. Our enzyme reaction is going to be dependent on shape. So as we investigate our proteins, we will see that their function will be critical to their proper, I'm not sure I use the right word there. So their structure will be critical to their proper function. And that is true especially with enzymes. There are some external conditions that can alter the shape of your enzyme. So we'll see that enzyme shape can be changed by temperature and it can be changed by pH. And so you have to have an optimal temperature and an optimal pH for enzymes to work. And for many cells, they are able to regulate those types of external features. If you're outside of that optimal condition, you can do a change to shape, which is called denature. Very often, that change of shape is permanent. So once you've changed it, you can't go back. Meaning, if the shape is changed, then the job is changed. So denaturing an enzyme will typically break it and it won't work ever again. Now, there are some proteins that can denature and then renature, meaning go out of shape and then go back into shape. So you will see that to some extent. Heating up an enzyme is probably the best example of a permanent denaturing of that enzyme. And you guys already know this. So you cook food, you can't make it raw again. You can't go back in time to that. And you know that when you cook food, it changes texture and color and flavor, right? That's because you've denatured those proteins. So a lot of those changes are due to the denaturing of those proteins. Okay, so let us go back to other functions that we see for our proteins. So in addition to this important job of proteins acting as enzymes, biological catalysts, we also see that some of the hormones that are used are made from proteins. We will see proteins involved as cell membrane channels and receptors. So all of this together is talking about ways that cells can communicate with other cells. We will see that proteins are important in the structure of our body, not just humans, other animals, not just animals, but across the board. So all organisms will have some structure connected into those proteins. This is at all levels. So if you're just a single-celled organism, then you still would have proteins important in the structure of the cell. We have proteins important in the structure of tissues. And then of course, organs are made from tissue. So we'll see some of our structure in our organs, and the whole body then is connected to those proteins. We have proteins that are involved in contraction. So when we talk about muscle for the body, we'll see that it's proteins involved in the contraction there. We'll see single-celled organisms have the ability to perform movement, and that's going to be tied to proteins as well. So we'll see that too. And then for animals, immune response is directly connected in with our proteins. So our antibodies are proteins, and then we have little name tags on our cells that allow our immune cells to recognize which cells are belonging to us and which cells don't. And those little name tags are composed of proteins. So we'll talk about these aspects a little bit more as we go through the class. Of course, some topics more than others. So we've already mentioned this idea that protein structure has a direct connection to the function of the protein. And I've mentioned already that the DNA molecule is the recipe or the directions for production of those proteins. And what it actually will tell you is exactly how to make that protein, meaning its structure. Additionally, we've mentioned in the context of enzymes, this is true for all proteins, that the environment can affect the shape of the protein as well. So conditions like temperature and electrolytes and pH can all, to some extent, alter the structure of the protein. And if you alter the structure, you alter the function. The reason that structure and function is so strongly connected here is that for proteins to work, they essentially have to recognize and bind to another molecule. So this ability to recognize and bind requires specificity of shape. So again, if you in any way change the shape of the protein, you change the function of the protein. So our next thing is to look at protein shape, which is fairly complicated, as you might imagine, since it's so directly tied in with function. But why do we need to talk through the protein shape? It has to do, again, with this connection of protein shape to protein function. Now, the complexity of protein shape has to do with the fact that there are 4 different levels of protein structure. So our first level we can call primary. So you can use your number 1 and then put your degree symbol at the top. The primary structure of a protein is a chain of amino acids. So amino acids are our building block or our monomer for protein. So of course that's something that we all need to memorize. When we look at an amino acid, we see that it is going to have an amino group. So that's a nitrogen with 2 hydrogens. We have just a lone hydrogen connected to the carbon. We have a carboxyl group, so that's how we're getting an amino acid. So the amino group and the carboxylic acid, the carboxyl group. And then we have what we have represented here as the letter R. So remember, we're using this R very generically, just like that X in algebraic equations can stand for any number. R is extremely variable. There are 20 different R groups, and here is a nice image taken from Google that is showing you the arrangement of the different R groups. There are 20 different amino acids that are used by cells to make proteins. So when we talk about the diversity of proteins that exist on the planet, this is another example of the monomers being the same, but the polymer being unique or diverse. So we can connect in these 20 amino acids into these wide variety of arrangements and give us different proteins. Those variations in R groups will be important because they will play a role in that tertiary or 3rd level of structure. So we'll come back and talk about that again. We are not, thank goodness, memorizing this chart. I just wanted to represent how R groups can vary, how much diversity there is in our 20. Going back to our discussion of our primary structure of proteins, it's a chain of amino acids. So those amino acids are connected together by what is described as a peptide bond. So if you have a chain of amino acids connected by peptide bonds, you can describe it as a polypeptide, where that prefix poly, of course, means many. So it's talking about many bonds. I am going to represent protein structure, a little hands-on activity. I'll bring this to class so you can play with it in class as well. So what I'm starting out with here is a pipe cleaner. It's nice and bendable. And I'm stringing my beads on there to represent those amino acids. You know, I don't have it completely covered with beads, so we're just getting kind of an idea or a feel for that. So we can think of our beads as those chains of amino acids connected by peptide bonds. That is our primary protein structure. Okay, so let's look at what comes next. Our secondary protein structure is coming from hydrogen bonding that's occurring along the length of the chain. So remember, hydrogen bonding is an interaction between or within molecules. So here we're looking at hydrogen bonding or interactions within the molecule. Now there's 2 different types of folding that can happen here. So I've represented already my my alpha helix. So it's just like a spring. So that's one way that our protein strand can fold. Another is described as the beta pleated sheet. So essentially what you see happening is molecules or components that attract each other are trying to position to be as close to each other as possible. Things that repel each other are trying to get away. So within the structure of that chain of amino acids, you're automatically going to get some folding happening. So this is the beta pleated sheet. This is my alpha helix. Okay. Okay, we're ready then for our tertiary structure. Tertiary structure, remember, is looking at those R groups. So now we get additional level of folding, again, more hydrogen bonding within the molecule. So here we're going to see hydrogen bonding between the R groups and the environment. Now remember that we're talking about proteins located as part of cells. So the inside of cells, and usually the fluid surrounding the cells, not usually, I think just exclusively, the fluid surrounding the cells will be aqueous based. Aqueous based means a watery solution. Water is polar. So remember that means that we have slight charges. So if we go back to our listing of our groups, we see that some are polar, some are non-polar. So if we stick these into water, it makes sense that you're going to have some hydrogen bonding or some slight interactions between those R groups in the environment. So this is the way I like to represent that. So I'm taking my secondary structure and then I'm just gonna sort of fold it up. Okay. Now, if I stick this in this environment, it might fold this way. If I change my environment, it might fold up another way. So the environment is playing a role in that tertiary structure. So when we mentioned that temperature and pH and electrolytes can all play a role in the structure of your function, structure of your protein, which of course relates to function, then it's this tertiary level that can be altered. I can also do that with this one. So just kind of random. Okay, so maybe it looks like this. All right. So now we're ready for our quaternary structure, our fourth level of structure. And this I like to just describe as a complex. So I'm going to represent my globular complex. Globular would be, oops, lost some parts. Globular would be if I take a beta pleated sheet subunit that has been folded, tertiary folding, and if I take my alpha helix subunit, and then if I bring them together, I now have my quaternary complex. I have individual subunits coming together. This one is the globular form because it looks like a glob, okay? Fibrous form would be more like a strand. So think of like hair or a fiber. So maybe it would have something more like this, a structure to it. Okay. Oops, sorry. All right. So here is a nice diagram. We have one that's similar in our book. This one was actually taken from Fundamentals of Anatomy and Physiology. So here you want to start by looking at your primary structure there at the top. That's, of course, looking at our chain of amino acids connected by peptide bond. This is determined, remember, by the DNA molecule. So the information is being directed here by your cell. And then your secondary level of structure is just going to be an automatic folding that's going to happen based on those interactions interactions within the molecule. So we see hydrogen bonding giving us our different either alpha helix or beta pleated sheet. The tertiary is going to be, remember, altered by the environment. So that's where we have that additional level of folding that can occur. And this is, of course, variable based on the environmental conditions. So in order to get it to be shaped exactly right, you need the exactly right environment. So sometimes if the environment is just a little bit less than optimal, then your folding will be a little bit less than optimal and it will still work, but just not as well. If that environmental condition is too extreme, too far away from our optimal conditions, then the folding will be such that it does not work at all. And remember, sometimes that can be a permanent change of shape, or sometimes it can denature and renature. And then finally, we have our quaternary shape. Here we're seeing, again, our globular form or our fibrous form. So there's a variation there. And that is it for now.